actually, as Hervé and also before him, Cédric and Marie stated, we got a phone call from Laurent asking us whether we would be keen on making a short summary of the past 20 years. And of course, I agreed. And actually, we both agreed that, so as you noticed, I'm quite young regarding to the other speaker and far less experienced maybe. So I thought that maybe I will tell you a sort of story depending on my own background and how actually I grew up within the field, because let's say I'm a kind of Mifobio baby. So not that I was conceived at Mifobio. I think those guys are still in high school. But I grew up as a scientist in here, and I'm really happy for that. And so maybe through this journey, you will have an overview of what's going on. So here you see a mouse embryo, regular, well, very standard imaging, bright field. And so the mouse embryo is actually around 80 microns. So here you see the cells dividing, organizing. It's surrounded by a small thin layer which is very transparent and protected, which is the zona pellucida. Then you may know this very famous movie of the early stages of the development of a zebrafish embryo. And here you can't really see it, but there is also this sort of surrounding transparent layer around, which is called the chorion in that case. And there was a new model that I discovered recently when I joined Pierre Nassois' lab. This is the encapsulated spheroid. So actually it's just one of the many ways of making balls of cells. And here the surrounding transparent shell is alginate, and we can tune the size of the ball depending on our microfluidic system. So the common features of all of those objects is that you have cells free floating at some point that self-organize and protected by this transparent shell, which is most of the time permeable to nutrients and so on, and also very conveniently permeable to light. So when you are a biologist and you address questions to those objects, of course, and if your readout of choice is imaging, you have to deal with at which scale am I going to work? So you may be interested in those objects in a protein-protein interaction, or to see whether bunches of proteins and submolecular aggregates would either are or even move. Or you can be also interested at the scale of the cell itself and have an overview of it. But you also may want to track and follow in time and in space a population of cells. And then if you go a step further, you can see all those individual behavior at the scale of the tissue, and of course, finally, at the scale of the embryo. So in order to access those informations, of course, you have to make choices. And this is a triangle that is very famous, and I like it very much because it provides you in a simple scheme that all the compromises that you have to make, because actually you cannot have it all. And actually there is even a hidden summit to that pyramid, which is the number of samples that you can image at the same time. So here everything is a matter of compromise. Depending on what was your initial goal, then you have to navigate through this parameter. Of course, if you want to have a very short temporal resolution, you may damage your sample, or and then damaging the state of your objects. So everything is a balance, and you have to navigate through that. So from the beginning of microscopy, so this is a bit of a silly overview, but what happens before us during the past centuries? So of course, you are all familiar with old-fashioned microscopes, which are really easy, and there is just one lentil and a sample holder and basically a screw, and that was it. But then people improved, of course, the light excitation, and so how to propagate light through the sample in a best way. And then during the 20th century, then it was an explosion of new technical developments, including the first, it's almost an idea without the possibility of making it, but from Marvin Minsky already thought about what could be the confocal microscope at that time. He just missed at that time the lasers that came a couple of years later, and then all together that enables the guys to build the first confocal microscopy, laser scanning microscopy. And then, as actually Hervé stated, then there was the reign at some point of the two-photon microscope that really it was a game changer in terms of depth penetration and all the new modalities that you can have. So when we start the 21st century, we can have a bunch of questions. And for example, what do we want next to have? Do we want to go deeper into the tissue? Because of course we are more and more looking at tissues that are scattering, full of lipids, and so it's really a nightmare to go through. Do we want to have more resolution? And this, as I told you, if you consider the triangle, then it might compromise the field of view or the length of the duration of the time lapse. Do you want to have a big field of view if your object is super big and you want to have it all? Of course, the question of having your sample healthy and alive is still at stake. And again, the high content. So I wanted to let you know as well that there are two round tables, because we do not have the answer, but collectively maybe we can address them. So I strongly invite you to join to those round table, because we need all of you, especially the physicists who are developing state-of-the-art imaging techniques that the biologists are not aware of. And reciprocally, I think the physicists need those challenging samples. So that would be a great occasion to discuss that and maybe to plan the future together. So to help the biologists to go through all the available technique, it's a bit of a jungle. You have heard many techniques. And actually, to make your choice, you have to pick according to a couple of parameters that you may be interested in. And then you have those useful, simplified, but still useful graphics that tells you, depending on how much in depth you want to go or how small you want to see the objects, then you can have a look and pick the microscopy technique that fits your experiments. It's a kind of tricky, but there are many reviews in the literature. This one is very new. It's a bit busy, so I won't spend time describing it, but depending on the scale, you have dedicated methods, imaging methods that indeed cover the light sheet, the multiphoton, actually, that I will stress a bit on that. And then you have also in parallel, the biologists and the chemists actually have developed many techniques to improve the labeling of the structure of the protein, of the interaction. And we are going also toward having some more functional markers in order to really be able to trace dynamic phenomena. And so I told you, amongst those lasers, I will those techniques. I will maybe stress a little bit more on light sheet, actually, not to give you a great insight in it, but just for the historical perspective. So we are lucky to have Jan Wisken here, and actually, if I'm not completely wrong, I think this paper was the pioneer one, where here you see the development of Drosophila pupa. And that was really a revolution, because the idea of having a perpendicular illumination and collection was not new, but applied to light and fluorescent imaging, it was really a sort of revolution. So here I like that, so it's in French, but I like this picture because it describes quite well, and with a hand that you may find in your room sometimes. So here it describes very well the comparison between the point scanning methods and the light sheet microscopy, meaning that, and it has already been said, that with those techniques you may bleach the whole object in depth, whereas with the light sheet you just illuminate one plane and then you can scan through it and even add the multi-view. So here, that's the only thing I would say, but I would also advise you to go attend, of course, to Jan Wisken workshop. There will be as well a main lecture by Boji Chung. And of course, I cannot be list everybody, but there are many other workshops and talks about light sheet microscopy. So that would be a great occasion to have an overlook of the new developments. And I wanted also, because by training I was a biologist, and I wanted also to stress that it comes from something that's not the best picture, but I think you can have an intuition of how messy it can be at some point. Of course, it's not messy, it's well organized. That was actually the first prototype that was built in Stelz's lab with Jan Wisken, that was transferred into a lab in the south of Paris, in bioemergence. And here you have the patent. So here it states that it was the first PCT was in 203. But then it has evolved in order to be more usable and commercially available for the biologists and other scientists. And here you can see the variety and the huge diversity of the setups that you can get, from something quite simple but very useful and versatile, including for big samples. The Zeiss Z1 that, of course, everybody knows, that is a box including everything, and especially the environmental control of the condition. Of course, the Luxendo and Viventis that are kind of close, and the phase view. And here I also wanted to show you Rémi Galland at work with aligning lasers, and with Jean-Baptiste they were building the SOSPIM that you may have heard as well. So the light sheet was really also a game changer, but as I started my PhD, I was interested in two photons. So I won't again go deeper into the technique, because so many other people really more competent in that will tell you about. But basically, indeed, you instead of having your continuous blue light to excite your favorite chromophore, you will use the two infrared photon at the same time. Plus, as Hervé stated, you can have an extra signal for free that is very useful and called SAG. And the principle actually is really intuitive, and the really great benefit is that you will excite only and bleach only the fluorophores that are at the focal point, where statistically this is the only place where you can have the interaction of the two photons at the same time, because of the pulse compression and the high numerical aperture, so high focalization of light into a tiny volume. And again, another benefit is that you will bleach your sample mostly on the plane that you are imaging, and as Hervé stated as well, you can go really deep into the tissue. Of course, excitation goes deep, but if you try to collect the fluorescence, it's a bit more tricky. But luckily, you know that all the photons that are emitted are relevant because they come only from the focal point. So if you improve the collection, then you can improve the quality of your image. So as I said, Hervé just talked about that, but there will be many talks, including by Marie-Claire Chanclin in the course of the module 5. And so I said that I will tell you a bit the story about the prism of my own experience as a baby Mifobio, let's say. So I did my PhD in Rennes, and I was at first interested in the development of the nervous system of the Xenopus laevis. And we were imaging them because we were lucky enough to have a 2-photon microscope in the lab. That's pretty much all we have. And I was recycling tubes and so on, but those samples and those microscopes were just enough to make me happy. And so we also were investigating signal for free. That was the idea of Denis, because at first I was only interested in the nervous system, but he said, oh, the myosin is giving such a great signal, we should investigate that. And that's what we did, and wrote a couple of papers with that. And so because we had that 2-photon microscope, I think we had some kind of, I would call the Maslow-Hammer syndrome. So the Maslow-Hammer is, for, if you don't know, if you have a hammer, everything is a nail somehow. So the joke here is that we were in Brittany, so actually we didn't image Queen Yeman, but we imaged some Madeleine and other crazy samples just to see, because starch actually provides some interesting signal. And my first Mifobio as a student was in Carqueiranne, and so we were only having posters, and I'm sure that the young researcher here, for the first time, you are open big eyes and just be amazed, and that was my case. I was very impressed. But actually, that's where I met not only my future thesis reviewer, but also my future first postdoc PI. And that originates because I saw that paper. Yeah, you know, you are a student, you do your bibliography, you are serious. And then that paper came out, and wow. And so just for you to have the wow effect as well, that was the main movie of the paper. So here you see the zebrafish developing with the THG signal that is roughly the membrane, the interfaces between the water and the lipid. And at the back, they projected the SAG signal. So here you see all the waves of divisions. And then they were segmenting the signal and extracting all the lineage from those cells. And for me, really, it was, yeah, as I said, the wow effect. And I got lucky to join, actually, Nadine Pereira's labs, where we had good time, and including the first Mifobio with them. And that was really an adventure. So we took the fish tanks, the water, everything to build the fish facility within site. There was here a really nice 2-photon microscope, and the guy from La Vision Biotech were super nice and giving us all the advices. We were injecting the same as we are doing right now. So for me, it's really nice. Laurent, yes, we were surfing at Talmont-Saint-Hilaire. I was not there. I think the water was pretty cold, and so that was the first crazy experiment that I saw there. And then, of course, in Nadine's lab, we were then applying all the techniques they have developed, and I was involved in a project that consisted in finding out where goes some cells that are actually in this structure at the back of the brain, well, the middle of the brain. These are stem cells, and so we wanted to know what happened to them and what structure they would colonize. And to the great disappointment, I think, of Nadine, because you saw previously that everything was automatized and everything, but here the cells were packed and crowded, and it was really difficult to image them. So I had, unfortunately, to do manual tracking. And so that was in 2013. Now things have improved because now we have other strategies to image the thing. Maybe you want to degrade the image, but go faster, and then you have more information, paradoxically. And then after Nadine's, I crossed the channel and was trying to apply the same techniques in Magda Jernicka's lab, and actually to mouse embryo. Then the level of difficulties were really improved because, of course, we didn't have such a microscope there. And so I had to discover actually the community of the guys involved in microscopy there. So in terms of network and really it was rich in terms of exchanges. I think Alex Sosik also came once to Mifobio to see what it was and was really impressed actually. And there, so I was then finally imaging fixed stuff and doing some 3D rendering. This is a human embryo that was cultivated in vitro. And then, so of course, the technologies evolved, and here was another striking paper by Kate McDowell. She was using a homemade SPIM with adaptive optics, and that was the tour de force of imaging the healthy embryo developing with multiple angles, tracking, and even you can follow the expression of some genes that are relevant for the biologists who are interested in the folding of tissues and the fate of cells. And so other people were then using also new light sheet microscopes that were more commercial at some point. And here are two examples. One movie? No. Yes. The first one comes from Iragi's lab. I think I saw it from the talk from Anna Kerschuk today. And so they were able as well to follow the development of the mouse embryo within with a light sheet microscope. And here is an example from Matteo Rodi, who is here as well and having a workshop. And I think it was really striking as well. But here we are dealing with really high-end commercial systems. So really sophisticated and expensive, of course, and you have to go to be serious about that. So that was the embryo part, which is one category of 3D samples. And the other one is organoids. So it started more or less, well, we can talk about semantics and history about that, but usually people agree on saying that it was a sort of invention from Hans Clevers. So here the very first proof of concept was collecting the crypts from mice, collecting the stem cells, sorting them, and then growing them in matrigel, and those structures magically somehow self-organize into those weird shapes with buddings, and actually those buddings reflect very much the organization of a crypt. And here you see a nice movie of the developing organoid. And approximately at the same time, the Madeleine Lancaster in the group of Knowledge, they were making those amazing mini brains. So of course, a beautiful picture like that is not completely the reflect of all the embryo organoids that they have in their dishes, but this one, you can guess that you have the cerebellum, the telencephalon, and all the layers that you can find in the brain. And they were even able to record live some spontaneous calcium activities within them. And then the thing is that in terms of imaging, what does that change to have those type of samples regarding the previous one? Because embryos, whether mice or zebrafish, they grow basically in salted water. They have all the nutrients inside as yolk, pellets, or a big yolk. But here, these are eukaryotic cells that you usually grow in 2D, and they require an incubator with 5% CO2 when you have it at 37 degrees. And you have to feed them with the DMEM and so on. But for them, it's an orgy, and it's an orgy as well for the bacteria. So really, it makes the experiments much more difficult. But people were brave enough to try. And here it's an example from the group of Prisca Liberali. And she was using this custom-made at first light sheet microscope. So they had to invent as well how to mount them, because those bunch of cells, they are very sensitive to confinement. So you cannot just include them into agarose, otherwise they will just don't develop well. And so here, by the end of the movie, you will see that you will have protrusions and so on. So they were really having some nice outcomes from the techniques, and then many papers to come with relevant biological questions. And then I wanted to come back to our module 5 actually, because this will deal with the imaging of 3D samples. So at first we will host Anne Rios, who is more interested in cancer organoids, and there will be beautiful movies as well. Secondly, Johan Spelleich, who is doing an amazing work on the morphogenesis of epithelium, mostly in Drosophila, but maybe he will tell us about more. As I mentioned earlier as well, Mike Lenchanclin will tell you about the multiphoton microscopy and will stress more on the methods. And this year we are also happy. It's been the second year we managed to invite someone involved in microfluidics and how microfluidics can help us to build 3D samples. So Mathieu Hautefeuille will give us a talk like that. I wanted also to remind the new thing, actually, the new thematic that Laurent said, that in the GDR, so we have this new group of interested on imaging the 3D samples. And here is an example made on site at Mifobio. And like the experiments we can do at Mifobio, you have the regular workshops, but then people gather, have ideas. I bring my sample, I bring my dye, I bring my knowledge in how to image the stuff. And here you have beautiful mouse intestinal organoids developing within the microscope. And this was possible because of the team. So depending on the year, we had, of course, Corinne Le Breton, Sophie Allard, Alessandro Furlan, of course, Lydia Danglo, Charlotte Rivière, who was supportive as well, and Gabrielle is also joining the group, Laetitia. And so now we can say that that's the rise of the OIDs in Mifobio. And so next, usually we are supposed to talk what's going on next, what's the future. And then maybe I will be, well, let's say provocative, but I hope not so. I would say back to the future from my perspective, but you will see. Actually, the spinning disk microscope was a little bit forgotten those years, but it has benefited from new developments in terms of light sources, the sensitivity of the cameras and so on. And so now you have many companies. Of course, the list is, so not only spinning disk actually, but all the sorts of confocal microscopy are now available by different companies, and they are cheaper than all the previous techniques that I've shown. So this is actually affordable within the lab. So you don't have to to have a true ERC to have a decent equipment. But then when your question requires those fancy techniques, actually you have to, and you are happy to benefit from the support from the platforms. And I think that's how it will work. Now, those fancy machines, not a single group can afford them because it costs a lot at buying, but then you have all the maintenance and so on. So we have, and actually that's what we do in the lab every day. We navigate through the simple devices that we have in the lab and the more sophisticated one that are on the platforms. And I think that's pretty much less. So on that, I will thank you very much for your attention.
Thank you very much for the nice talk. I was wondering about the temporal scales for the development of these organoids.
Yes.
So because when we are developing techniques, it's always nice to know. Thanks.
So I will maybe answer you by being both from Normandy and a biologist. It depends. So as I said, it depends on whether you are interested in a rapid interaction between proteins that will maybe like kiss and run, interact and just and go away, or in more physiological point of view, when you are a pathway that is activated, then it has to turn on, act, and then be turned off, and then could be in the range of hours maybe. But then when you are more interested in morphogenesis, again, it depends on the structure. Some structures are very quick to set up because the cells proliferate quite fast. So then maybe for a day or two. But then, for example, for bigger organoids or big spheroids, and depending whether you just want to monitor the growth, then the trick is to have them under the microscope for weeks sometimes. And then you really have to change actually the way of thinking on how to design. Of course, booking a 2-photon microscope for 2 weeks, I cannot afford it. So actually in the lab we developed what we called an incubascope, which is a very basic microscope that permits us to follow the organoids developing for weeks. That is very cheap, and actually it depends on the readout. We were interested in monitoring the shape and making segmentation of them, and for that application it's enough. So really the technique, it depends on what is the readout and what is the information you have to extract, and I think that's the very first start of the experiment.